All right, moving on to now the last part of what I want to communicate. And again, this is a strategy in terms of how we can make use of what we do well as breeders and the emerging data. I've hammered home the point quite a bit about population size being important. And after Walter's talk, there were several questions about analysis of the data and experimental design that lead me to think that the potato community is thinking about population or experimental design in a very positive way. So one of the things that we've done to maximize population size through experimental design is shifted some of our work to augmented designs. So for those of you that may not be familiar with an augmented design, I just want to give you a quick overview. Basically, the way an augmented design works is that we create our population is essentially unreplicated, right? But we replicate checks in such a way that we can account for different kinds of spatial variation in the field. And so in this particular design, we're accounting for variation. Let's see if I can get this mouse to go. We're accounting for variation in rows, in columns, and also in quadrants. So we have 4 rows, 4 columns, and 4 subquadrants, right? And so we can then, and then the X's are marking the spot for the replicated checks, but everything else is unreplicated. Now, the reason that this can work when we're doing analysis is based on markers is because we're going to collapse it down anyway, right? It's the genotypic class that has replication, right? The actual line itself doesn't have to have replication. It's the genotypic class that we need to have replication on, okay? And so we use the overly replicated checks to account for field variation and make corrections as necessary. And so in this scheme here, if we had a randomized complete block design, we could handle 72 individual genotypes. If we use an augmented design, the way I've laid it out here, we can look at 128 genotypes. Okay, it's much more powerful from the perspective of looking at those marker trait linkages to have that level of genotypic replication. Okay, so larger populations but equal resources, and we can account for field variation. Okay, and so the data would look something like this, and I've already put in an example of a row and a column normalized, but you don't have to do that yet. Really, the first step is to ask, is there spatial variation in the field? And so what I'm showing you here is the results of an analysis of variance, and what you see is that, yeah, there's significant row within location variation. Okay, so we know that we have to correct for that. And so we can normalize our data for that row variation and then rerun the analysis. And what we see is that when we do that, that variation goes away because we've normalized our data to account for it. Okay? And so all of the significant effects are now removed except for the genotypic variance, which is what we're interested in as breeders. Okay? And so the idea is that we first run the analysis to ask, is there field variation and what accounts for it? Then we make the correction, and that it's that corrected data that we then would use for our integration of genotype and phenotype. Okay, so what I just showed you was an example that was based on considering the genotypes in our populations to be fixed. But I would argue that we might actually want to consider them to be random. They are random selections out of an infinitely large potential population. And if we are going to consider them to be random, then we need to be thinking about a different way of treating the data. And this schematic, by the way, is directly from a classic textbook on variance components, and the reference is there for you. Okay, and so if we consider the genotypes to be random, we need to estimate our traits based on a best linear unbiased predictor. And we can get estimated BLUPs from the proc-mix procedure of SAS very efficiently. There's 2 different ways of doing it that are shown here. The output of these 2 different ways is slightly different, but you can get what you want. And the upper method where we're just putting the solution, the slash solution, takes a lot less coding. The bottom method, we get the actual estimated BLUP, but takes a lot more coding to do. Okay, and so from that upper method, we get an estimate that's relative to the mean of the population, and then we can adjust. But the point I'm making is that we can take an augmented design trial, estimate the BLUPs, and then use that as our trait data for the marker trait analysis. Now, what's important to realize is that when you start to build up data sets over time that have overlapping sets of genotypes, is that you can estimate a BLUP for that whole data set, right? And so even though you don't have the same genotypes every year in the trial, you can begin to merge data, right? So the best linear unbiased predictor essentially allows you to predict the performance of a line that wasn't in the trial, in the same way that we can predict the milk yield of a bull, right? Okay. So this slide really sort of ties it up, and I don't want to go through all the details. What I really want to emphasize, so what I've done here is I've kind of color-coded some of the different aspects of a program. So the red represents analysis that gets done. The green represents really the activities of our breeding program in terms of making crosses, planting material, and collecting data. The blue represents the genotyping component, and then the yellow selection. And what I want to emphasize is that marker-assisted selection is going to be most powerful when we can use it to shorten the breeding cycle, and that's what I'm trying to show here. So if we can use it in this context to shorten the breeding cycle, over time we will make more progress. Okay? And so in conclusion, as breeders, we still need to recognize the importance of measuring phenotype. Point of analysis is going to be an important concept to graphs, so we're not necessarily going to be working with single SNPs, but pairs of SNPs, so haplotypes. Incorporating pedigree data or population structure into analysis improves the detection of trait associations and also the efficiency of marker-assisted selection. Our power goes up with population size. The number of markers we need really depends on recombination. Finally, phenotypic selection is effective. We know that, so we can't abandon the resources that we put into that. Mass can help if we use it to reduce the cycle.